Can you hear me? Yes, okay, thank you. Hope you have a very great day. I hope you have a very great day. We are starting our lessons. Do you have any questions for the previous session? Do you have any question for the previous session? And one of you, please tell me, where did we finish? Good morning. Good morning, all. We want to hope that you will stay for the lecture channel date. Only if it is suit Dr. Emily put it on Sunday. We have 4 lectures on Sunday. Fatima, I'm so sorry. I couldn't. I can't because today this is my only free day, and we have classes, and they replace it with molecular immunology. They replace it for molecular immunology. So I don't have time. I'm so sorry. If you can have the class today, you can ask. I choose any time that suits you so that we can be with you. How many classes do you have today? How many classes do you have today? One? Just this one? After this, do you have any other class after this class? No? Okay. Let me talk. Let me talk to the university. So if we can, I can make another class after this class. Okay, I will let you know. I don't promise. Okay, I don't promise, but let me talk about it. Okay, thank you. Thank you. Fatima, do you remember where did we finish? I love you too. Thank you. It's my honor to teach you as my international students. It's my honor. On page 28, okay, so we finish here. I didn't tell you this. No. I think we were here, yes? We were select right. We were here. Yes, here. Okay. Any questions up to now? Any questions up to now? Okay. Let me repeat. We were talking about the DNA extraction. Okay. We were talking about the DNA extraction. I told you in the previous session that we culture the bacteria in the Bros agar. I told you we have two kinds of Bros agar. M9, I told you defined and undefined. Do you remember? Defined and undefined. For example, M9 or LB, as you remember. After that, I told you, after we grow the bacteria, we centrifuge it. The place of bacteria settle down. We have bacteria here. Look. We centrifuge the bacteria, so we have plates of bacteria. How we can centrifuge? We can centrifuge at 800 RPM for 10 minutes. Okay? After that, we want to prepare cell extract. Marve told us, how can we prepare cell extract? Two different ways. One is chemical ways, and one is, I told you, physicals. Thank you very much. So we have chemical ways and physical ways. Okay, what's about the physical ways? Do you know? Can you name some physical ways to break the cell walls, to break the bacteria? Tell me the physical way to break the bacteria, break the cell walls. Can you? Temperature, yes, Nata, yes, thank you. If we boil the bacteria, so one way is boiling. What will happen if you boil the boiling? One way is boiling. If we increase the temperature, the cell wall will break. So one of the physical ways is to boil. Okay, and decrease the temperature. So if we freeze the bacteria, so freeze and thaw is the other way. So one way is increasing the temperature, for example, boiling, and one decreasing the temperature, freeze and thaw. What else? What else can you say? What else? Tell me about physical ways to break the bacteria. I told you about increasing the temperature, like boiling. What else? No, Fatima, at lysosome is chemical way. We are talking about the physical way, you know? We don't want any chemicals. We want physical way. For example, decreasing the temperature, increasing the temperature. For example, we have some UV eradication. Yes, we have some free radication. Or what else? We can destroy the cell walls by let me find the words. Do you know, for example, if you want to talk about the plants, if you want to talk about the plants, as you know, the plants are so very so firm walls. So what do they do for the plants? You can pound them. You can pound them. You can ponder in the, for example, you can break the cell walls. Okay, after that, we have chemical ways. So let me talk about chemical ways. As Fatima told us, we can add some enzymes. For example, what? Lysozyme. What does lysozyme do? What does lysozyme do? Sorry, it's lysozyme. What does lysozyme do? The lysozyme will break the cell walls. So lysozyme can break the cell walls, and we can add some chemical like EDTA. EDTA. What is the EDTA? EDTA is the substance which remove magnesium ions. Okay, EDTA or the EDTA removes magnesium. What does magnesium do? Magnesium plays an important role in a structure of cell wall. Okay, if edit or remove magnesium, what will happen? The structure of cell wall will drop. So the cell wall can break. So EDTA of, yes, thank you, Mr. Haydar. Remove magnesium. The magnesium, they play an important role in cell walls. Okay, for example, this is cell walls. We have some Mg here. We have some Mg here. The EDTA removes magnesium. Okay, they removed the Mg. So if EDTA remove the Mg, the structure of cell wall drops. So the cell can break. The other role of EDTA is it can remove magnesium too. Again, and what does magnesium do? They are cofactor of DNAs. Look at here. What does DNAs do? What does DNAs do? Please tell me. What does DNAs do? The oxyribonuclease. DNAs. What does DNAs do? Tell me. DNAs break the DNA. Okay? DNAs break the DNA, RNAs break the RNA. So, if we have DNAs, there is some Mg in its structure. It has some Mg in its structure. If EDTA remove the Mg, the DNAs can't work properly, so it can't break the DNA. It can't break the DNA. Understand? So EDTA can do two important roles. They can remove the Mg. The Mg are in the structure of cell wall, so the cell wall drops. The second role is it removes the Mg from a structure of DNAs, so the DNAs can't break the DNA. So we can add EDTA and lysozyme here. Magnesium, why is it there? Where? We have Mg everywhere. In your body, we need Mg everywhere. We have Mg in the structure of cell wall, and we have Mg as a cofactor or coenzyme of DNAs. Okay? Is it a part of solution or the composition of DNA? No, it's the part of a DNA polymerase. Do you remember DNA polymerase? DNA? Remember? DNA polymerase? Yes? The structure of DNA polymerase needs Mg. So many enzymes. For example, DNAs needs Mg. So many enzymes in their structure needs DNAs. Okay, Mr. Zaid Razak wants me to repeat the second role. I told you, in the second role, it can remove Mg too. So we have Mg in the structure of DNAs as cofactor of enzyme. So if the Mg removes, the DNAs can't work. If the DNAs can't work, it can't break the DNA. Understand? Here we want DNA. If DNAs break the DNA, so we don't have DNA. We want DNA. We want to keep it. We want to protect the DNA. Understand, Mr. Zaid? Okay. So here we use EDTA and lysozyme for breaking the cell walls. After that, we want to break the or we want to lyse the membrane. We want to lyse the membrane. What can we add to lyse the membrane? What can we add to lyse the membrane? As you know, membrane is made up of lipids. Yes, membrane are from lipids. If we add SDS, the SDS lyse the lipids. It's one kind of detergent. Do you know detergents? It's one kind of detergent, so it can lyse the membrane. So for the membrane removal, we can add SDS. We can add SDS. Do you understand up to now? Okay, so up to now, we break the cell walls and we lyse the membrane. How we could break the cell walls by physical and chemical ways? For example, for chemical ways, we could add lysozyme and EDTA, and for the membrane, we could add what? Some detergent like SDS. So we can have cell extract, and we have some cell debris. You know, this is the cell walls. We have some cell debris. How can we separate the cell debris? We can centrifuge. So after we break, we can centrifuge. If we centrifuge our microtube, the cell debris go down. The cell debris go down because they are heavy, and we have some DNA, RNA, and protein in the supernatant. So in the plate, we have cell debris, and in supernatant, what do we have? We have DNA, RNA, and proteins. Okay, look at here. Now, okay, let me explain more. So the next step is that we want the supernatant and we don't want the cell debris, okay? So we can take the supernatant, and there is some contamination here. Do we want proteins now? Do we want proteins? Tell me, do we want proteins? To extract the DNA, do we want protein? No, we don't want. We just want the DNA. Do we want RNA? No. So what should we do? We should omit, we should clear the proteins, and we should clear the RNA, and can prepare the pure DNA. Okay, so listen here. We should omit and we should clear the RNA and proteins, and we should prepare clear DNA. How? We have two different ways. The first way is degradation of the contamination. And the second way is separation of DNA from contamination. So the first way is to degradation of contamination. How can we degrade the contamination, for example? Can you tell me? No, this one. I'm talking about this one. In this step that we want to degrade the contamination. How can we degrade the contamination? For example, we want to clear proteins. How can we omit the proteins? For example, we can add protease. Yes, Fatima, thank you. We can add protease. Yes, protease. How can protease do? They can omit the proteins. So in the first step, we can add some enzymes. For example, protease and RNAs. Protease can clear the proteins. RNAs can clear the RNAs. So we have pure DNA. Okay? In the second way, look at here. We separate the contamination. We separate the contamination. How? Look at here. Look at here. Let me look at here. If we want to separate the contamination, this is the cell extract. We can add phenol. We can add some phenol and chloroform. We can add some phenol and chloroform. In the portion of 1:1. In the portion of 1:1. What does it mean? It means I add 1 ml of phenol and I will add 1 ml of chloroform. Okay? So 1:1 phenol and chloroform. After that, I make the microtube up and down. I vortex the microtube very slowly. I up and down the microtube. Up and down the microtube, and then centrifuge the microtube. Centrifuge the microtube. After the centrifuge, we have 3 layers. Look at here. We have 3 layers. In the upper layer, it's aqueous layer. We have DNA and RNA. Look. In the middle layer, we have some proteins, and in the bottom, we have phenol. So, if we I told you, if we centrifuge, after the centrifuge, we have 3 different layers. Chloroform is here. Did you hear about chloroform? Do you know the chloroform? Yes. Okay. So, did you understand? Did you understand this? Okay, thank you. So, this is the phenylchloroform, and so which part do we want? Which part? This is 3 layers. Which part do we want? This is in the book, Fatima. You can read the book, and there's very good explanation in the book. So which part do we want? We want upper layer. So we use the upper layer. Here is DNA or RNA. If there is more contamination, we can repeat this step one more. So if there's still contamination in this, we can repeat this step one more. But be careful, because the DNA could break. So it's better not to centrifuge again. One other way you can do. So if there is still some contamination, you can repeat all this step again, but it can break the DNA. So what can we do? For example, we can add some portals here. Why we can use some portals? Because the portals here don't break the DNA. So if there is still so much contamination, we can repeat or add some portals to the DNA and RNA level. And how can we omit the RNA? How can we clear the RNA? Tell me, please. How can we clear the RNA? What can we use to clear the RNA? Add RNAs. Thank you, Mr. Natak. We can add RNAs. Okay? No question up to now? If you don't have any questions, let me talk about ion exchange chromatography. So we want to separate the DNA from contamination. Okay, so listen to me. We have to separate the contamination. This is the ion exchange chromatography. What does it do? It has very small granules of silica. So it has very small granules of silica which has the positive charge. So very small granules of silica has positive charge. What's the charge of the DNA? Negative. Thank you. So the DNA has the negative charge attached to the silica which has the positive salt. So look at here. This is the column which has the silica with positive charge here. We add the cell extract here. Okay, the cell extracts go down. At first, some substances which doesn't attach to a positive charge go out. Then we out some salt. We add some salt. After we add some salt, the particles which doesn't attach so firm to the silica go out. For example, proteins and RNA. And after that, we add more salt. The DNA which attach so firmly go out. So look at the picture. You can see at the first step, we centrifuge. We add cell extract. And what do we have in the column? We had a silica which has positive charge. The first step, the particles which doesn't attach to a positive charge go out. The second step, we add some salt, sodium ions. The particles which attach not so firmly go out. And the third step is the DNA which attach so firmly go out. Understand? Okay. Thank you. So up to now, we culture the DNA, we culture the bacteria, we break the cell walls, analyzes the cell membrane, and we prepare the cell extract, and after that we purify the cell extract. How could we purify the cell extract? We had two ways. We clear and we purify the cell extract. And what do we have? We have DNA. We have DNA now. But now we want to concentrate the DNA. We want to concentrate the DNA. How can we concentrate the DNA? Look at here. Here we can, again, two ways. We can do it in two different ways. Look at here. In the first way, here is DNA. We can add some ethanol. We can add some ethanol. Ethanol should be 100%. So the ethanol should be 100%, and it should be cold. So we add cold 100% ethanol to the DNA. Okay. After that, we can add a glass rod. The DNA fiber will bind to glass rods. So this is the first way. It's not a very good way. Why? Because it may the DNA may become contaminated if we put a glass rod our DNA may become contaminated. The second way is better. Look at here. In the second way, again, we add some ethanol. I told you again, it should be 100% and it should be cold. Okay? It should be 100% and it should be cold. After we add ethanol, we shake it very slowly. We shake the microtube very slowly. What should we shake the microtube very slowly? We should up and down the microtube or shake it very slowly. Why should we shake it very slowly? Don't break the DNA, thank you. Because it may break the DNA. So we add some ethanol, 100% cold ethanol, and then we centrifuge it. We centrifuge it in very high speed because the DNA is very light. The DNA is very light and very small. So we centrifuge it in high speed. After the high speed centrifuge, the DNA precipitated, and we have ethanol in the upper in supernatant. We remove the ethanol, we don't want it, and let it dry. Sometimes we upside down the microtube to let it dry. Because we don't have ethanol here. Do we add ethanol twice? Yes, sometimes. Sometimes we add ethanol twice. Do you know why? Because if we think there is any contamination here, is here, we add it twice to clear the contamination. The second step, we can use the 70% ethanol. 70% ethanol. So the first step should be 100% cold, and the second step you can use 500%. Okay? Understand? So for the DNA concentration, we could use two ways. Which was better? The second one is better. Thank you. The B was better. I told you we add ethanol 100% cold, and the DNA we centrifuge. I told you we remove the ethanol. We upside down the microtube to dry the ethanol. Okay? Who knows what is the next step? What is the next step? Do we use a sediment of the DNA? or we should use it the liquid form? Did you saw the DNA in the laboratory? They're all liquids, so can't use the DNA like this, which is sediment. Okay, we should add. So after the ethanol dry, we should add some liquid. Or some solution to solve the DNA. What can we use? We can use some water we call DDW. Very sterile water. I think it's an Arabic name, taktir. Taktir is Arabic name, yes? We use it. Okay. This water, taktir twice, okay? So we use DDW. DDW is the water which taktir twice. Understand? Or we use TE, sorry. We use TE. Would you please let me a second? I was talking about adding some solution to the sediment of DNA. I told you we can add some DDW or TE. Do you know the difference? In TE, we have tris EDTA. So we can use both. For example, sometimes we use DDW and sometimes we use TE. TE has tris EDTA. So do you know, anybody knows the difference? Anybody knows the difference? No? And what's the beneficial of each? So what's the benefit? For example, if DDW is better, tell me what's its benefit, or if the T is better, tell me what's its benefit. This is not chemical, so it's better. Which one you mean? DDW. No, Fatima. T is better. Okay, I told you. T is better. Why T is better? Because in TE, the lifetime of DNA is more. So the DNA can stay longer in TE. So if we add some TE, the DNA can stay longer. Understand? What is DDW? I told you, Astra, you didn't. I told you TE, the DNA which we use TE, which we solve it in TE, can stay longer than DDW. So the lifetime of DNA which solved in TE is better than DDW. Understand? But why? I think none of you knows. So please search the reason. Please search the reason for the next session, okay? So in the next session, please tell me why DNA stay longer in a TE. And please remind me that I ask you this question. Okay. After that, how can we store the DNA? How can we store the DNA? After that, we can store it in the freezer. We can store it in the freezer. I mean, in the temperature is minus 20 degrees or less. So we can freeze it in minus 20 degrees or in minus 80. So we can freeze it in minus 20 or minus 80. Okay. No question? Okay. If you don't have any question, please listen here. Please listen. Look at this picture. Look at this picture. In this picture, I'm talking about another way of DNA purification. Another way of DNA purification. What's the beneficial of this method? For example, if some cells has so many contamination, for example, they have so many carbohydrates, or they have so many proteins, so they have so many contamination, it's better to use this way. So if our cells has so many carbohydrates or so many proteins and have so many contamination, it's better to use this method. Can you please tell me which cells has so many carbohydrates? So many carbohydrates, do you know? For example, plant cell. If we have some plants, as you know, plant has so many carbohydrates and so many, for example, proteins. So we can't use the previous methods that I told you. We can use the CTAP methods. Look at here. Look at here. This is the CTAP methods. So in this method, we use a substance called CTAP. It can name as cetyl trimethyl cetyl 3-methyl ammonium bromide. So cetyl 3-methyl ammonium bromide. Let me write it for you. Cetyl 3-methyl ammonium bromide. It's CETA. Look at this. This is the cell extract. This is the cell extract here. After that, we add some CETAP. What does CETAP do? CETAP bind to nucleic acid. Bind to nucleic acid. Look at here. Look at the picture. After we add to CETAP, CETAP add to nucleic acid, and they become a complex, and the carbohydrates become soluble, become so much soluble, and they break. So after that, if we centrifuge CETAP and DNA or nucleic acid go down, and after that we can discard the upper or supernatant. In the upper layer or supernatant we have so many carbohydrates and proteins. We discard the supernatant and we add 1 molar NaCl to the plate. Why should we add NaCl to the plate? Because we want to remove the CETAP from the DNA. So we remove the CETAP from the DNA. After that, we want to concentrate the DNA. How can we concentrate the DNA? I told you in the previous, how can we concentrate the DNA? We could use ethanol precipitation. We add 100% of cold ethanol and centrifuge. Okay? If we have some RNA here, how can we get rid of some RNA? How can we get rid of RNA? We can add RNAs, thank you, or ribonuclease. And after that, we could add some TE. We could add some TE. And we have pure DNA, and we can store it in freezer in -20 or -80. Understand? Do you want me to repeat more? Okay, look at here. I told you if there is some cells which has so many contamination, like, for example, plant cells. In plant cells, we have so many carbohydrates, yes? For example, tell me about rice. What do we have in rice? So many contamination, so many carbohydrates, yes? Or for example, maize. We have so many carbohydrates. So if some cells have so many carbohydrates or so many proteins, we can use the method CETAP. CETAP presents as cetyl methyl ammonium bromide. This is one substance. What does it do? It will bind to nucleic acid. So look at here, the blue one. Bind to nucleic acid and deform the other substances. For example, deform the carbohydrate or protein and make them soluble. After that, we centrifuge. If we centrifuge, I told you CETAP bind to nucleic acid, yes? CETAP and nucleic acid go down. So in our plates, what do we have? We have nucleic acid and CETAP, and all others are in supernatants. All others in supernatants. Do we want supernatant? Do we want the supernatant? No. We should discard the supernatant. So we discard the supernatant, and after that we add 1 molar NaCl. We add 1 molar NaCl. Why should we add 1 molar NaCl? To separate the DNA from a CETAP. Okay, so after that we add 1 molar NaCl to separate the DNA from CETAP. If we have some RNA here, what should we do? I told you it binds to nucleic acid. We have DNA and RNA. Okay, if some RNAs, we can add some ribonuclease to omit the RNA, to clear the RNA. After that, we want to concentrate the DNA. What can we use? We can use ethanol precipitation to concentrate the no, Mr. Haidar. 1,1 was for phenochloroform, not here. Phenochloroform, we don't use it here. After that, we solve them in TE or DDW. We solve them in TE or DDW. There minus 20 freezer. Okay. Okay, thank you. So it was so easy. Thank you. Okay. Please look at here. Please look at here. Here is guanidinium thiocyanoate chromatography. So the next way to separate the DNA when we have so many contamination, for example, we have so many carbohydrates, or we have so many, for example, proteins, we can use guanidinium thiocyanoate columns. Okay. Look at this. Look at this. Look at the picture. When we add, look at this. We had one silica which had positive charge. I told you the DNA has the negative charge, so they bind to silica particles, and after we add salt, we can separate the DNA from the silica. Okay. Now we add some guanidium thiocyanate. We have some guanidium thiocyanate. We add the cell extract with guanidium thiocyanate. In the guanidium thiocyanate, when we add guanidium thiocyanate, all others except DNA denaturate. So all the biochemical denaturate except the DNA. Okay? If we add guanidium thiocyanate, all the biochemicals, for example, proteins, carbohydrates, all chemicals denaturate. So they go out from the column and we can discard them except DNA. Look at here. We have some silica particles. We have some silica particles here. We can have some silica particle which have positive charge. Okay? We add guanidium thiocyanate. Guanidium thiocyanate breaks and denaturate all other chemicals, but DNA attach or bind to silica particles and they don't go out. After that, we use some water and we can see the DNA go out. Okay, understand? Look at me. So, it's so easy. Why? Because guanidium thiocyanate denaturate all other substances except DNA. When we add cell extract, all others, for example, carbohydrates, proteins, RNAs, all denaturate and go out except DNA. After that, we add some water and DNA go out. Okay? Thank you so much. You want some rest? Okay, if you're tired, have rest for 5 minutes. 5 minutes, I mean. Okay. Again, can you hear me? Hello again. Okay, you want me to be your teacher for روش تحقیق? Okay. If you ask Dr. Maqami for being my student, she can prepare a class for me today. You can come to my class. I tell her too, but you can tell Dr. Maqami too. Okay? Okay, thank you very much. Please do not forget this. Okay, Fatima. After the class, I will tell her. Any questions up to now? Any questions up to now? Why did we add salt at the first time and the second time we add water? No, we just add water here. Iman, look at here. We just add water. In the guanidinium thiocyanate column, we just add water. That was the ion exchange chromatography that we had 2 steps of salts. Look here. It was ion exchange chromatography, Iman. We add salt, and the second step we add more salt. But here in the guanidinium thiocyanate column, we had silica and we had guanidinium thiocyanate. At first, all the carbohydrates and protein denaturate and go down. If we centrifuge, all the substances except the DNA go down. Okay, so we don't add anything. All go out and we discard this. After that, we add water. And after we add water, the DNA separate from the silica. Okay, Iman? Understand? Thank you. Let's talk about preparation of plasmid DNA. Let's talk about the preparation of plasmid DNA. So, as you know, look at here. If this is a bacterial cells, we have some plasmids and we have bacterial chromosome. Look at this. Can you tell me in this picture, or as you know, what is the difference between bacterial chromosome and plasmid? What's the difference between bacterial chromosome and plasmid? Plasmid is circular. Bacterial chromosome is circular too, Fatima. Both are circular, but plasmids are small, or very small. Yes? They are very small, circular double strands, and the chromosome of bacteria are so much larger. In the so we can separate the plasmid one by their size. So we can separate the plasmid one by their size. And what else? We can separate the plasmid by their conformation. Shapes. Mr. Natek told us their shape are different. Do you know what's different in conformation? So I told you two different ones. Their size, we separate them by their size, and we can separate them by their formation. Okay, listen. The first I'm talking about separation of plasmid by their size. Separation of plasmid by their size. So, the plasmids are very small circular DNA which are in bacteria. Okay, which are in bacteria. How can to separate it? Look at here. Look at here. At first, we add lysozyme. What did the lysozyme do? Do you remember? Lysozyme, I told you at the beginning of the session, lysozyme break the cell wall. Yes, break the bacterial wall. Okay, look at here. At first, we add some lysozyme and EDTA. As you know, as I told you, lysozyme with EDTA break the DNA wall. But we have something else here. What do we have? We have 25% of sucrose. Okay, we had lysozyme and EDTA plus 25% sucrose. What does it do? What does it do? It doesn't let the cell wall to denaturate, and it can make the spheroplast. I think you know about the spheroplast. So if we add EDTA and lysozyme with sucrose, it prevents the cell from bursting immediately. So a spheroplast will form. So the cells with partially degraded cell wall that retain an intact cytoplasmic membrane are called spheroplast. So after that, as you remember, we want to break the membrane. What did we use to break the membrane? Remember, in the beginning of the sessions, we add SDS. Do you remember? I told you this is the detergent. But now we don't add SDS. We add Triton Xad. Why we use Triton Xad? X100. I'm sorry, I use it Persian. Triton X100. Why do we use this? Because this is non-unique detergent. SDS was unique detergent, and unique detergent breaks the DNA. But Triton X100 is non-unique detergent, which doesn't break the cell walls. Okay? After, if we centrifuge the cell extract, cell debris which are very heavy go down, large DNA fragments of the bacteria which are large and heavy go down too, and the plasmid which is small and so light are in the supernatant. Understand? I repeat very quickly one more. Okay? Listen to me. Be careful. Listen to me. We want to separate the plasmid. Plasmid are so small. What is the difference between the chromosomal bacterial chromosome are very heavy and this is so small. We add lysozyme, EDTA with sucrose. If we add sucrose, the DNA wall breaks partially and the membrane are intact. We call it a spheroplast. So membrane is intact. We have the membrane and the cell wall disrupts. We call it a spheroplast. So we want to prepare a spheroplast. After that, we add Triton X100. Triton X100 is detergent, but non-ionic detergent. Non-ionic detergent doesn't break the DNA. So the chromosomal bacteria which is heavy never breaks. So we centrifuge, it bind to cell walls and it's heavy and doesn't break and go down because they are heavy. And in supernatant we have plasmids